Welcome, everyone. We are going to wait just a couple seconds to let everyone into the Zoom. While you're joining, please feel free to pop your name and location in the chat. Say hello, let us know where you're joining from. We always love to see who's with us. Okay, well, I am Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. And welcome to this November 2024 webinar presented by the ASCCT and the European Society of Toxicology in Vitro. I want to encourage all of you to visit our websites to find out more about our organizations and becoming a member and signing up for our newsletters to support programs like this webinar series and some of the awards that we give out to early career and young scientists. This webinar is recorded, and the recording will be posted on the ASCCT website a few days from now. In the webinar platform, you can ask questions of the presenter using the Q&A feature in the toolbar at the bottom of your screen. There will be a Q&A period after each presentation is finished. So while our speakers are talking, please feel free to put in your questions in the Q&A. And you can also vote for questions that you really like. And finally, we've enabled closed captions, and you can turn them on or off in the toolbar. So I wanted to give you a couple of reminders. First of all, the ESTIV Society has launched its campaign for 2026 meeting, which is going to be in Maastricht in late June and July. And they are asking for session proposals. So please go to their website if you'd like to participate and put in a session proposal. Also, we have a webinar coming up in a couple of weeks on December 4th. This is a partnership with the Japanese Society for Alternatives to Animal Experiments. And the speaker will be Dr. Kaori Ambe, Associate Professor at Nagoya City University, and she'll be talking about the prediction of skin sensitization using machine learning. If you want to know all of our upcoming webinars, because in fact we're scheduling quite a few of them for the 2025 first few months, go to our website. If you just go to the homepage, you can see this little tool on the right side that shows all of the upcoming webinars and events. We have ASCCT and ESTIV webinars, as well as some other partner webinars that you might be interested in learning about. So make sure to stay up to date and register for those webinars. Okay, so we'll get on to today's presentations. First of all, Eliška Kuchovska is a postdoctoral researcher at the Leibniz Research Institute for Environmental Medicine in Germany, specializing in the development of human-relevant, animal-free new approach methodologies to evaluate the effects of chemicals on brain development. Driven by her fascination with the complexity of the brain, Eliška combines rigorous research with enthusiasm for science communication. She actively promotes career development for early career researchers and serves as the chair of the ASPIS Academy and the EUROTOX Early Career Forum. She's going to be presenting Ontology-Based AI-driven innovative approach using DNT NAMs for NGRA. And this presentation was a winner of the Best Oral Presentation by JSAAE. And the second presenter is going to be Tiago Marques Pedro, who earned his Bachelor of Science in Biomedical Science at the University of Surrey, UK, which included a research placement year in toxicology at Weill Cornell Medicine in New York. He went on to pursue a master's in toxicology at Karolinska Institute in Stockholm, where he explored the effects of environmental pollution mixtures on genotoxicity and the use of DNA repair inhibitors to combat resistance to cisplatin-based chemotherapies across various research projects. Currently, he is a third-year PhD student at the University of Cambridge, and his project focuses on using machine learning to predict mitochondrial toxicity. This presentation, which will be using machine learning framework to improve the efficiency of mitochondrial toxicity screening by guiding compound selection, was also a winner of one of the ASCCT-ESTIV Best Oral Presentation Awards. And without further ado, I will stop sharing and turn the floor over to Eliška.
Born to the US. Let me share my screen and find the laser pointer. Yeah. Thank you so much, Christy, for the nice introduction, and also thanks to Ellison for organizing the webinar. And I would also like to thank the Japanese Society of Alternatives to Animal Experiments for awarding me for this presentation at the ESTIV Congress earlier this year. And today I will present the same presentation that I presented at the ESTIV conference with a few small updates. And my work actually is in the framework of the ONTOX project. And as already introduced, the talk is entitled Ontology-Based AI-Driven Innovative Approach Using Developmental Neurotoxicity New Approach Methodologies for Next Generation Risk Assessment. Well, the title is quite long, but we will cut it into pieces. And first of all, I want you to focus on the keyword of developmental neurotoxicity, because this is where our study actually comes in. We are all, as you all know, we are all exposed to various chemicals every day that may cause harm to human health. And this is also true for prenatal exposure to chemicals, which may then cause developmental neurotoxicity, short DNT. Well, there are hundreds of thousands of chemicals on the market, but only for less than 200 of them we know if they cause the DNT potential, the DNT effects or not. Well, this is caused by the fact that currently the DNT is assessed following several guidelines, especially this OECD guideline number 426. And this guideline employs animal models, especially rodents, such as rats, for testing the chemicals. Well, unfortunately, these tests are very expensive. It costs around 1 million euros or US dollars to test one chemical. It's also very time demanding. We need more than 1 year to test one chemical, so it's definitely not high throughput and fit for purpose for the vast chemical universe that surrounds us. And most importantly, what should interest us as scientists is that the animal models are not really well representative of the human health. There are numerous species differences, especially in the brain development. For example, different temporal patterns, different liposomes, different certain molecules that affect the brain and regulate the brain development. And simply the extrapolation of results to the human situation is definitely not easy. So because of this, I think that we really need this paradigm shift and adopt new approach methodologies that are human-based and fit for purpose for the human situation that we are interested in. And these new approach methodologies can be either in vitro models, human in vitro models, or for example, in silico computational predictive models. Well, and this is exactly what the ONTOX project is working on. Our goal is to create new approach methodologies that will provide a functional and sustainable solution for advancing human risk assessment of chemicals without the use of animal testing. And we divided our work into 3 tiers. And this workflow starts with data collection. So we collect data from different domains like biology, chemistry, kinetics, and toxicology, anything relevant for the risk assessment of a chemical we are interested in. And we integrate this data into a framework that we call an ontology. You can imagine it as a multidimensional framework that integrates all these vast amount of data, but then it's quite difficult to filter through the data and find what you are interested in. So that's the reason why we have it linked with an AI system that can help us see and filter through the data. Of course, there are some data gaps because what we collect from the literature won't be complete for all the chemicals that we are interested in. So we have it linked with a battery of in vitro assays that we use to test the chemicals and to fill then the gaps in the ontology. Of course, this, together with exposure assessment, then leads to the final next generation risk assessment that we want to do. And this new innovative approach we aim to implement into legislation. Since I talked about the in vitro batteries, I would like to briefly just show you which in vitro assays we have in this in vitro battery in the ONTOX project. If you are familiar with the OECD-supported DNT in vitro battery, our first model is the neurosphere assay that employs 3D neurospheres to mimic some of the key neurodevelopmental processes that need to happen in a precisely orchestrated manner during the brain development. Our second cell model is an iPS-derived cell model that is used to assess the synaptogenesis key neurodevelopmental process, and this method is developed at the NIPH laboratory in Norway. And our last assay is the neural network formation assay that employs iPS-derived neurons and primary astrocytes and was developed by my colleague Christina Batman. Today in this presentation, I want to talk mainly about the first year data collection, but also to show you how an ontology actually looks like, and also about the in vitro tests. And I want to jump right into the what we call a first pillar of the ontology, which is the physiological map. Physiological map is quite an innovative tool. I'm not sure if you are familiar with it. It's like a knowledge base or a framework that integrates physiological but also homeostatic processes of a certain organ, so in our case, the developing brain. And this is only on the molecule and cellular level. And how we create such a map? Well, we do a literature review. The results that I will show you were done using an old school non-systematic manual literature review, but we also used AI supported tools for this literature review, but maybe we can show you the results some other time. So today, results are done using non-systematic literature review following standardized physiological map guidelines. And then together with my colleagues from the Université de Liège, the results are mapped in a software called Cell Designer and uploaded onto the Minerva platform, which is an interactive web-based platform when you can play with the data that we collected. So the resulting physiological map of the developing brain is actually multi-layered because of the brain complexity. So the first layer is composed of 4 different submaps, and each of these submaps focuses on the development of a different cell type, so neurons, astrocytes, oligodendrocytes, and radioglia. The second layer is the layer focusing on cell-cell interaction, so the most important interactions between the cell types that I just mentioned. And finally, we have an overview to guide the reader through the map. Most importantly, this map is 100% human-based because, as I already mentioned at the beginning, we don't want to bias our map with the numerous species differences that are there in the brain development. So we used only data based on human iPS-derived cell models or primary cell models and data measured on human fetal brain samples. So without further ado, when you go into the Minerva platform on the interactive webpage, first thing that you can see is this overview, which shows you which kind of cell types, which key neurodevelopmental processes are covered by the map. And as I said, it's interactive, so you can click on any of the cell types or processes, and it will redirect you directly to the part of the map you are interested in. The second layer is then the cell-cell interactions. You can see the main cell types present during the brain development, and you can see, for example, which proteins they excrete that then influence, for example, activate or inhibits any processes, or how it influences other cell types during the brain development. But I want to spend the most time on one of these submaps. So this is an example of the oligodendrocyte submap. It's the most complex one that we have right now and most detailed one. And I want to point out that it was created with my dear colleague Louis Ladeyrav from the Université de Liège. And this map, you can see that it's very nicely structured. So let me walk you through the map. So if we look in the first section here called the inputs, we see different proteins, for example, here the FGF, the fibroblast growth factor protein, that then in the second section of the map on the membrane binds to different receptors, for example, the FGF receptor here. If we go further through the map, here we have a section of intracellular mechanisms. So the FGF that bound to the FGF receptor activates a cascade, in this case, the FGF signaling pathway, until we reach the end of the map. And here in violet diamonds, we have the key neurodevelopmental processes that are linked to the different cell types that are at the bottom of the map. So you know that this map is focused on the development of oligodendrocyte cells. So we start here with neuroprogenitor cells that differentiate into oligodendrocyte precursor cells that differentiate into premyelinating oligodendrocytes until we reach the final stage of myelinating oligodendrocytes. And the whole map then shows you which proteins or which signaling pathways are important, which are regulating these key neurodevelopmental processes that these cells undergo. So for example, we can see which signaling pathways activates or inhibits the different processes. And the nodes on the map have either a special color or a special shape, depending on what they are exactly. But today, I just want to highlight the ones that we have here in orange, because I told you previously that we created this map based on a literature review, but these orange ones, they are actually integrated into the map via a different way. So these were created based on our experimental studies that we did with one of the cell models that I presented previously. So we used the neurospheres in the neurosphere assay to see which signaling pathways regulate this key neurodevelopmental process in the neurospheres to later integrate the data gaps in the physiological map. And this work was done in cooperation with my colleague, Christina Bartman. So if you are familiar with the OECD-supported DNT-IVB, this assay is one of the DNT-IVB assays. It's called the NPC5 assay and assesses the oligodendrocyte differentiation. And so what we did, we exposed the neurospheres to specific inhibitors and activators of selected signaling pathways that you can see here in the outer ring. So for example, we used celecoxib, which is a specific inhibitor of the cyclooxygenase 2 pathway, and we observed that it decreases dose-dependently the oligodendrocyte differentiation. We did this with all these signaling pathways, so now we know which pathways regulate and which not, which decreases oligodendrocyte differentiation, which increases the oligodendrocyte differentiation, and we thus have an overview of the biological applicability domain of this assay. Well, since the neurosphere assay can mimic different key neurodevelopmental processes and not only oligodendrocyte differentiation, but also, for example, neuronal differentiation or the migration of different cell types or the proliferation of neuroprogenitor cells, we did this with all these signaling pathways and all these assays to establish the biological applicability domain of the whole neurosphere assay. We did this not only to fill in the data gaps in the physiological map, but especially to decrease the uncertainty or to increase regulatory confidence in this assay. Most importantly, we also wanted to increase the human relevance of the assay. So for this purpose, we did another literature study focused on neurodevelopmental disorders related to the signaling pathways and to the effects that we observed in the neurosphere assay. So if I come back to the example of the cyclooxygenase 2 pathway, I found that the cyclooxygenase 2 pathway is impaired in the neonatal white matter injury. And one of the hallmarks of neonatal white matter injury is also disrupted oligodendrocyte differentiation. So by linking what we observe in our assays and in the neurodevelopmental disorders, we increase the human relevance of the assays for regulatory purposes. Now again, we did this for all the assays and all the signaling pathways. So we came up in the end with a lot of data on neurodevelopmental disorders that we wanted to show in the map as well. So we called for help to the Minerva team, and they kindly created a plugin for us to show these neurodevelopmental disorders on the map. So when you go to this webpage platform, you can actually search, type in any neurodevelopmental disorder you are interested in, or you can type in oligodendrocyte differentiation or the FGF protein. And if we have a neurodevelopmental disorder linked to it, it will be shown on the map. So to sum this part up, we created a multi-layered physiological map of the developing brain that is 100% human-based. And as I already said, it's mostly a knowledge base, right? But actually, physiological maps are an excellent tool with multiple purposes. So let me show you. It doesn't serve only as a knowledge base. You can also use it to derive new in vitro assays for your batteries. So for example, you know that this part of the map you already have covered by an in vitro assay, but there is a small part here that you don't have covered, and you know that you should develop a new in vitro assay to have your in vitro battery that can assess all the key neurodevelopmental processes that are present in the developing brain. You can also use it to derive new adverse outcome pathways. So obviously, for example, for a new key event in your key event sequence, or also to help with the assessment of biological plausibility when you assess your key event relationships in the adverse outcome pathways. Another, and this is the most exciting and my favorite purpose of physiological maps, is the creation of predictive in silico models. So imagine that you can parameterize all the nodes on the map in a software, and then in a Boolean approach, you can play with it and switch on or switch off the inputs that you have here at the upper part of the map. And the software will then predict if the key neurodevelopmental process that you are interested in, so on the bottom of the map, if it will happen or not, if it will be inhibited or not by the inputs that you switched off or on. And finally, as I said, the physiological map is the first pillar of the ontology framework that is the center of the ONTOX project. But how exactly does the ontology look like then? Well, to make it easy, you can imagine it as a simple overlay on the physiological map. So for example, we are overlaying the physiological map with transcriptomics data. So then it looks, for example, like this. And we overlay it with different types of transcriptomics data. So first type is healthy transcriptomics. For this purpose, we downloaded datasets from the Brainspan Atlas, which is an excellent resource of actually amazing datasets spanning from different developmental stages, different brain regions, different individuals of fetal brain samples. We also did the literature study on neurodevelopmental disorders, as I already mentioned, and also found a linked transcriptomics dataset. So we plan to overlay these disease transcriptomics datasets over the physiological map as well. And finally, to make a link with our in vitro models, we also overlay the transcriptomics of our in vitro models on the map. So for example, the Neurosphere assay that I already mentioned, it was already published by my colleagues in 2021. But I am actually very excited that a few days ago, I think 2 days ago, we published a new transcriptomic characterization of our iPS-derived cell model. So if you want to check out the publication, I will leave the QR code on tomorrow's slides as well. Besides transcriptomics, we want to overlay the physiological map also with physicochemical data in cooperation with other partner of the ONTOX project. And finally, we also overlaid with an AOP network. So this AOP network, we compiled based on 16 adverse outcome pathways from the AOP wiki, but also scientific literature. We also manually curated the network. And as you can see, we are interested in 2 adverse outcomes of interest: decreased cognitive function and impaired learning and memory. So you see that we are still going for the human relevance, still going towards the neurodevelopmental disorders. And this is actually what we did earlier this year. We published a publication where we linked key events from the DNT AOP network to neurodevelopmental disorders. And we also pointed out any key event and disease gaps in the AOP wiki. So to sum this up, we created the physiological map. We have the basis for our ontology, but I told you that we want to do risk assessment, right? Well, this definitely wouldn't be enough for risk assessment. But ONTOX is a big consortium where each of us do our small part. So if we combine our in vitro batteries and the physiological map and the ontology with other aspects like quantitative in vitro in vivo extrapolation, physiologically based pharmacokinetic modeling, the quantitative or read-across structure-activity relationship, If we add the quantitative aspect to our AOP network, and of course, the exposure assessment, we can do the next generation risk assessment we were aiming for. And for the implementation, then we are, of course, in active communication with stakeholders. This year, we started doing a case study of this probabilistic risk assessment using PFOA. So I think that next year, stay tuned for more results about this. To conclude my talk, I believe that we have a solution for a safer human brain development that we are developing in the ONTOX project. And this solution is innovative, thanks to the physiological map, the ontology framework that is linked to the new approach methodologies. This solution is also highly ethical because we save life of animals. We also decrease the costs of the test, the time requirements, it's more high throughput. And finally, the solution is sustainable and reliable and can enable us to do the probabilistic next generation risk assessment of chemicals. And I would like to thank you very much for your attention, for being here today. But especially I would like to thank my co-authors of this work and also our IUF team led by Katarina Koch. So if you have any questions, I would be very glad to answer them, but you have also my email here to send me any questions later if you want. And you can also subscribe to the ONTOX newsletter to stay updated about our news. Thank you so much.
Thank you very much for that presentation. I have to say I came up with a couple of questions and then you answered them. One of them was about case studies because I'm kind of involved, or at least monitor the OECD activities on developing the in vitro battery. And I know that there are plans to put together some more case studies. So it's good to hear that you're working on that.
Yeah, sure. We need case studies to persuade the people that it makes sense, that it's working, of course.
Yeah. Okay, so first question coming in, and let me just tell the folks listening online, please put your questions in the Q&A. We'll ask them. Did you use artificial intelligence models to generate the submaps? And if so, which models did you use?
Okay, so far we did not. At least the results that I was presenting, it was a manual literature review. But my colleague Luis Ladeira, that I also mentioned during the presentation, he is now using new software, but I don't remember the name, unfortunately. And the goal is now to update the oligodendrocyte submap with this result. So I know that he screened many papers using the AI tool, and there are indeed many notes that he can add to the map. So in the publication that we will submit very soon, and which will have this version of the map, it will still be done using only the manual curation. But for our next paper that will be led by my colleague Luis, it will be already with the AI supported search. So, or just drop me an email and I will put you in contact with Luis and he will tell you which software uses.
Oh, and that's great. And I can actually tell you that Luis presented in this series a few months ago. So if you're interested, you can go to the webinar archive and learn more about that work as well. I'm wondering also, you talked a little bit about linking or using physiological maps to create AOPs. Can you talk a little bit about how that works? I mean, I know that it seems like the physiological maps are more detailed than an AOP in the typical wiki. So is there some process of like determining which parts of the maps are key events and which aren't, or kind of how does that work?
Yeah, that's very difficult because you don't want to have too many key events either, only the most important ones. The important thing is that the maps are only on the molecular and cellular level, so it would be only the upstream key events, first of all. And we are actually now in discussions with the SCIG group of the SAAOP, and we will present about it in December. I don't remember, at the beginning of December, they have a meeting and we will actually discuss the use of physiological maps for the AOP development. Great. Together with the AOP specialist. So if that person who asked the question is interested more in it, just send me an email And again, I can put you in contact. But so far, I would say we use it only as a knowledge base. So you have a look in the map, and that guides you if you missed anything important, for example. But for example, for the biological plausibility assessment of the key event relationships, we have to discuss it first with the AOP people, and that will be discussed at the meeting at the beginning of December. But I think it's really promising.
Okay, yeah, that's great. I should mention actually that the SAAOP is creating a partnership with ASCCT and ESTIV, and so we'll be having a lot more events that are related to, and the SAAOP is a Society for Advancement of Adverse Outcome Pathways. So we can probably share what comes out of that meeting for sure with our membership. Yeah, this is kind of a related question. I'm going to take one more question and then we'll see if you can answer some of the questions in writing format so that we can get on to the next presentation. But this one is, do you have any plans to extend the AOP to see the correlation with morphological features in the brain?
So the AOP network that we built integrates the AOPs that were already published. We are also developing new linear AOPs, and we are doing a new one on oligodendrocyte development, for example, and another one on proliferation linked to microcephaly, if that answers the question. But the AOP network itself includes just what's already published. So our development of linear AOPs is another task, but yes, we are developing new AOPs as well. And maybe I can invite people here. So if there are any students who are developing AOPs and maybe are a little bit lost, at SCCT in Switzerland, when Ellen Fritsche is now based, we recently founded, it's called an AOP hub, where we, actually it's a group of students and a few senior AOP people, and we are answering the questions if they have any about AOP development and guiding them how to develop it. So again, if you would be interested in joining this group, just send me an email, and yeah, it's kind of friendly and nice group, just helping each other how to build AOPs.
Great. Thank you again, Dr. Kus. I'm going to try it.
Kuchovska.
All right, we're going to move on to our next presenter. Tiago, would you like to share your screen?
How does that look?
I'm seeing the outline.
The outline, okay. Does that look better?
Yep, that's the full screen.
Okay, brilliant. Yep, so hello everyone. My name is Tiago. I'm a current third-year PhD student at the University of Cambridge in the UK. I just want to firstly say thank you to ESTIV for awarding me the oral prize at EUROTOX 2024 in Copenhagen this past year, and also to inviting me for this ASCCT-ESTIV webinar here. And today I'll be giving a talk on an aspect of my project where I utilize the machine learning framework known as active learning to guide compound selection to improve mitochondrial toxicity screening. To begin, I want to just provide a bit of background. So as we all know, the cost of developing new drugs, therapeutics, is very expensive and it takes a very long time. On average, it costs $2.6 billion and takes 12 years to develop a new therapeutic agent. And these resources and the cost of these resources are exacerbated if we have any adverse events with these medications. And a lot of these adverse events are due to human toxicity, which is a main cause of almost 50% of all attrition cases for these therapeutics. A major player in this is mitochondrial toxicity. And here we have a few examples of drugs that were released on the market for different purposes, and all of them led to side effects such as cardiotoxicity, due to mitochondrial tox. And more specifically, in each of these cases, it was due to inhibition of an electron transport chain, or ETC complex. Now, this sets up nicely to frame the problem that we are facing and also the main aspects of my PhD project. We have off-target mitochondrial toxicity, a major contributor to this being drug-induced ETC complex inhibition, and also the fact that many of the toxicity prediction methods that we currently use lag behind the development of new chemicals and drugs. This could all lead to potential adverse events, and in most severe cases, drug attrition. And as mentioned already, this is not only a loss to the resources, the time and money put in, but most importantly, it means the potential loss of life-saving therapies and medications to the hands of patients. So I'm going to focus a little bit on the toxicity prediction methods. And traditionally, when we want to screen a chemical library, we will take each compound and screen them sequentially until we've completely screened that library. Now, this is fine when the library is relatively small, when you don't have that many compounds to test. But what happens when you have a much larger library? It would be useful to have a tool that could help guide you to select what order you should screen compounds and when, in order to return the toxic chemicals sooner. Now, this was a predicament I found myself in during my own project, where I had an assay, a tool to screen mitochondrial toxicity, and I had quite a large library. So to talk a little bit about that technique I wanted to use to screen for mitochondrial toxicity, I used the glucose-galactose ATP assay. So the main aspect of this is we took HepG2 cells, so these are cancer cell lines derived from hepatocellular blastoma cells, and due to the Warburg effect, they rely heavily on glycolysis for energy production, but they prefer this over oxidative phosphorylation for the production of ATP, because glycolysis also produces other useful resources such as nucleotides to allow for cancer proliferation. So in our HepG2 cell, we have glycolysis running, production of ATP, and if you pair this with an assay such as cell titer glow luminescence, this utilizes the ATP produced by the cell for the luciferase reaction. And through that, there's an emission of light, which you can measure as luminescence, and it gives you an indication of cellular viability. Now, you can switch this glucose sugar source for galactose. And what we see now is the cell is unable to utilize glycolysis to the same degree that it usually would to produce ATP. And it is now utilizing oxidative phosphorylation for that production of energy. And this makes the cell very reliant on mitochondria, as OXPHOS occurs in mitochondria, and now the mitochondria is key for producing ATP. And again, if you pair this with the same assay, that emission of light is now more indicative of mitochondrial function. So when you pair this assay with glucose and galactose cultured cells, it gives you an indication of the cytotoxicity of a chemical, but also the mitochondrial dysfunction. So I had this assay now, and I wanted to screen the Prestwick chemical library. This is comprised of 1,520 small molecule compounds, 98% of which being FDA and EMA approved drugs. And I am, only one PhD student. I thought to myself, I really wish I could make this a lot more efficient. So that's where that idea kind of sprang from, and it's where the concept of active learning comes in. So with any traditional supervised machine learning model, we took known compounds and chemicals that are mitochondrial toxic, so they're known to be toxic to the mitochondria, and also chemicals that are known non-mitochondrial toxic. We took their structural features, which are encoded as fingerprints, and we then used those fingerprints to train a machine learning algorithm. In this case, it was a random forest model. Now we take that trained model and we use it to predict the Prestwick chemical library for mitochondrial toxicity. However, we are also assigning each of these predictions an uncertainty score, and this is calculated based on entropy. Now, with this, the prediction predictions can be ranked based on how uncertain they are. So most uncertain predictions ranked highest, and you can take a subset of the most uncertain predictions to be labeled with their accurate activity labels. And this is where you can either try and look for any existing data on these chemicals, whether you can find evidence of mitochondrial toxicity and assign labels that way, or it allows you to experimentally test these compounds. So you can take the most uncertain chemicals and actually physically test them potentially with this technique that I mentioned, the glue-gal ATP assay. And this will allow you to assign a specific activity label, whether the chemical is toxic or non-toxic, and you can add this back into that original training data. And you can repeat this process over several iterations, hopefully with each iteration improving on the last until you've screened the entire Presswood chemical library. Now, the point of this would be you wouldn't have to screen the entire library to be sure that you've identified nearly all of the toxic chemicals. That's what this workflow would hopefully help to provide. So with machine learning models, you want to be seeing what you're putting in, what you're training the model with. So to start, that initial model that I built, that was comprised of toxic chemicals from the TOX21 mitochondrial membrane potential assay, or MMP. So they identified over 1,000 actives or mitochondrial toxic chemicals. And for my non-toxic, I looked at a chemical database. So I gathered oral safe drug compounds with no mitochondrial mechanism of action. And also I removed any chemicals that had hepatotoxic or cardiotoxic alerts. In addition, we ran a literature search where if there was any hits that went against the search terms of mitochondrial toxicity, those were also removed. And this was all done to ensure that we had as promising of a data set that was not mitochondrial toxic. We also did some further processing to handle any duplicates. So if there was if there was a chemical present in both our toxic and non-toxic data set, we made sure that it was classified as toxic because it at least showed there was evidence that it could be mitochondrial toxic. So that was a more conservative measure and a way to ensure that we weren't missing any potential positives. So the structural features would be encoded in fingerprints of this initial data set, and those fingerprints would be used to train a random forest model. And as mentioned, that would be used to predict the Presswood chemical library, ranking the predictions based on uncertainty using entropy, as we can see in this calculation here. Now, it's always good to visualize your chemicals in the chemical space. And here we're looking at toxic chemicals and non-toxic chemicals, so toxic red, non-toxic in blue. And this is the initial training data. And this UMAP represents, is encoded using structural features. So the distance between data points represents structural similarity. And the main takeaway is we're seeing a slight separation between our toxics and non-toxic chemicals, but there's no clear-cut clustering. And using a cheminformatics software such as Data Warrior, we can identify specific clusters. And I showed this to you in the 3D space because these clusters could be missed if we were just looking at a flat 2D space here. But those same clusters identified in the 3D plot, we can see them here represented. So here are some toxic clusters and their representative chemicals, and also some non-toxic. We can also do this now with comparing that initial training data and also the Presswood chemical library, which is the data set we want to predict, we want to test. So that active learning initial training data is in teal, the Presswood chemical library in purple, and any chemicals present in both data sets are in the gold-orange color. And again, this is based on the chemical space. So these are structural features, and distance represents structural similarity. And the main takeaway from this plot is that our training data is very structurally similar or inhabits a similar chemical space to the Presswood chemical library, which is what we want to predict. And so hopefully this training data should extrapolate quite well to predicting the Presswood chemical library. And again, same as before, we can identify clusters, and we can see how that's represented in this 2D plot with representative chemicals. Now, it was decided that due to the burden of preparing and aliquoting the library for screening, we would run the entirety of the Prestwick Chemical Library's Mitochondrial screen first, so all biological runs, and then we would implement the active learning framework to see if that screening could have been improved. So after our 3 biological runs, we had initial classification of these chemicals. So we're looking at the cellular ATP levels in galactose cultured HepG2 cells on the x-axis, and the ATP levels in glucose cultured HepG2 cells on the y. We identified 102 toxic chemicals. So these were chemicals that reduced ATP by at least 30% in the galactose cultured cells, but not in glucose cultured. And that's important because if we see reduction in glucose as well, that means that it could the reduction in ATP could be due to cytotoxicity and not specifically mitochondrial toxicity. And so this inconclusive set, these 109 chemicals we're seeing in orange, they did exactly that. They reduced ATP in both galactose and glucose cultured cells. And so we could not distinguish whether this was due to mitochondrial toxicity or cytotoxicity. For this set of inconclusives, we took them forward and we ran a dose response experiment to assign accurate activity labels. So once we did that, we came with a final classification of 1,404 non-toxic in blue and 116 toxic chemicals in red. Now, this plot on the right is slightly different to the one on the left. This one is, again, another UMAP based on structural features. So the closer they are, the more structurally similar they are. And as you can see, we have a lot of red points very close to our blue ones, meaning that there is no clear gross structural difference between our toxics and our non-toxics in the Prestwick Chemical Library. And this could pose a problem for when we use our model for predicting these chemicals. Additionally, I thought it would be interesting to look at any chemicals that were present in both the Tox21 and also the Prestwick Chemical Library datasets, and see if any chemicals that were toxic in my screen of the Prestwick Chemical Library, if they were also toxic, those same chemicals in the MMP assay in Tox21. And from this table, you can see that all chemicals that were present in both datasets, the ones that I found toxic were also found toxic in the MMP dataset, and the same for non-toxic chemicals. So there was 100% concordance between the two assays, and it just adds hopefully a little bit of weight on the evidence, not only on how I ran my screen, but also that we have a lot of this public dataset seems to be quite reliable as well, and it hopefully complements one another. We also ran an enrichment analysis on the results of our screen. So the main takeaway here is that the descriptors here on the right were significantly enriched in the mitochondrial toxic class compared to the non-toxic class. And it just opens up another area of investigation of why is this the case? Why are these descriptors enriched in the toxic class? So how will we actually assess these mitochondrial toxic hits? Well, as mentioned by Eliška, this is not as complicated of an AOP, but it's one that describes the process of liver injury due to mitochondrial toxicity and potentially specifically due to ETC complex perturbation. So this is a published AOP on the AOP wiki. And if we look at this and use this as almost a backbone and a framework as to where we are on the chain, we've currently looked at mitochondrial dysfunction using the glugal ATP assay. But with each key event, we could identify potential experiments that we could use to determine whether our chemical is actually causing this key event in this AOP pathway. So for example, for looking at binding to the complexes, we can use computational techniques such as molecular docking, but there are also specific assays that allow us to look at different site inhibition. So for example, with complex 1, you can determine whether it's the FMN site or the Q site. As mentioned before, we looked at general mitochondrial dysfunction using the glugal ATP assay, but now we will also look at specific mitochondrial respiration using the Seahorse platform, where we can measure extracellular flux. Intracellular ROS plays a massive role, and also the timing of when ROS occurs can play a major factor into how the chemical is actually causing toxicity. And finally, also looking at the type of cell death. And we'll take all of this battery of assays and use a triaged approach. So we will take all the hits through first, and then gradually that number will drop as we identify different hits at different endpoints. And this was a strategy that was taken by a previous PhD student within our lab, Rachel Hardy, and she used this exact same triaged approach to assessing third-generation antipsychotics to see if they were ETC complex inhibitors. But for the big question, the main essence of this talk was about whether active learning could have improved the efficiency of this screen. So what actually happened? Well, when we used active learning, we saw differences in several performance metrics, but the one that really stood out was positive predictive value. Now, this is the ability of the model to accurately predict toxic chemicals. Here, we're comparing it to random selection. Random selection is more reflective of traditional screening methods. This is where there's no uncertainty-guided approach to selecting which chemicals you should choose next to screen. This is just done at random. So the solid orange line represents the average of 10 runs of random selection, and the shaded region is the standard deviation across those 10 runs. And it's being compared to active learning, which is in blue. The message here being that when we screened only 40% of the library, we were confident in 92% of the predictions of toxic chemicals, compared to only 61% in random selection, potentially offering a framework and a workflow that could be a lot more rapid in screening and identifying your toxic chemicals sooner. And it now opens up questions about what is causing this uncertainty. Could it potentially be structure? Now, what we're seeing here in this plot is a activity cliff analysis. This was again done in Data Warrior. So each point denotes a different chemical in the Presswick chemical library. Again, distance reflects structural similarity. So you have more similar chemicals closer together and really similar chemicals in these networks that are joined, as you can see here. Also, we have mitochondrial toxicity depicted based on color. So mitochondrial toxic chemicals being red and non-toxic being blue, light blue. And as you might have noticed, we have chemicals that are very close to one another, but of different colors. And these are known as activity cliffs. In this example here, we have clindamycin, which was non-mitotoxic in our model and in our screening, and lincomycin, which was mitotoxic. And as you can see, they have an almost identical chemical structure, aside from a change in a functional group. Now, this is not a be-all, end-all demonstration of a flag of, okay, if a chemical has a change in this functional group, the same as here, it's going to be mitochondrial toxic. That's not really the case. It's very contextual, it's context heavy, but it opens up a really interesting case study as to why we're having such a different effect on activity when there's such a small structural change. So to summarize, we found that active learning was able to predict mitotoxic compounds with over 90% accuracy when screening less than half of the library. And I believe the framework could benefit not only in resource-limited situations where maybe you don't have a lot of time, or maybe you cannot afford to buy an entire chemical library, but also if you want to prioritize the identification of toxic chemicals and assess those first. And now with this, we want to explore a bit further. So I showed this plot before where this is our chemical space of our training data, our original training data, and our Presswick chemical library. And I said that they extrapolate well because they inhabit a similar chemical space. But what happens then if we try to use our framework for chemicals that inhabit a different chemical space? And it's here that we want to explore now. We want to verify the utility of the framework on these uninhabited of chemicals in this different chemical space and see whether our model can still produce the results it showed. With that, I'd like to thank everyone for listening and also thank everyone that helped on this project, McFarlane Lab, and also all the other contributors at the MLC Toxicology Unit. Thank you very much, and I'm happy to answer any questions.
Wonderful. Thanks, Tiago. Got a couple of questions coming in. The first one was they were looking for a reminder. Did you mention, how did you distinguish between general cytotoxicity and mitochondrial toxicity?
Yeah. So with how we did the metabolic switching, the HepG2 cells usually utilize glycolysis, and this is through the sugar source glucose. But when you remove that sugar source and you give them galactose, now they're mitochondrial dependent because oxidative phosphorylation is the key driver of creating ATP, of generating energy. And so what we did is when we expose the cells either cultured in glucose or galactose to a chemical, if they reduced or if they caused a reduction in ATP in the galactose cultured cells, but not in glucose cultured cells, it was an indication that there was a mitochondrial effect occurring due to that chemical. If we saw a drop in ATP in both glucose and galactose cultured cells, you can't really distinguish whether that drop in ATP is due to mitotox or due to cytotoxicity.
Next question. They are talking about the idea that mitochondrial electron transport chain inhibition can be a wanted pharmacological activity for certain antifungal drugs, crop fungicides, biocides. Do you see an evolution of your human system based on HepG2 data with limited metabolic competence to additionally inform species selectivity and de-risk compound pipeline for these cases? And there's a couple of follow-ups, so I don't know if you want to open the Q&A and keep it in your mind or answer that part, and I'll ask the additional questions.
Yeah, maybe just to not maybe mess up the sharing and so on. Yeah, so yeah, that's a very good point. I think there's one of the big weaknesses of this technique using the glugal ATP and HepG2s is, again, as mentioned, the lack of CYP enzymes, which means that you're not getting your metabolites. And as we all know, if you have generation of potentially toxic metabolites, that could be the main driver of toxicity. And this is something that I'm trying to currently address. There's different ways you can go about it. Either trying to predict the metabolites using machine learning, or just try and identify the metabolite structures using chemical databases online. That is probably a better case. However, if we took those metabolite structures and then we used our model trained on glugal ATP to predict the activity of those metabolites, because of phase 1 metabolism leading to the metabolites being very similar in structure to the parent compounds, it's very likely that any predictions of the metabolites will have the same activity as their parent compound, which could just add a lot of noise to the model. So this is an element I'm thinking about. But in terms of species differences, this is kind of why I think the activity cliffs are really interesting, because those two compounds that we saw, they are of the same therapy, or they're the same class of compound. And yet one is very, very toxic to human liver cells and the other one isn't. And I think this work, maybe it's not good, it won't provide a universal substructure or toxicophore, but I think it could provide a bit more insight into why we may see these dramatic changes in activity.
Yeah, and maybe almost following on from that, they're asking how you feel the system would deal with intrinsic assay variability versus potential species differences.
Well, so what I found was interesting was it took me 3 months, maybe 4 months, give or take, with things in between, to run all 3 biological repeats and of the library, and also the dose response of the inconclusive chemicals. But the model, using the uncertainty guidance, it was able to predict toxic chemicals quite well. If in all honesty, it was quite good in that role, which was quite surprising. So I think that was quite a big benefit with using this model. I didn't have to run it several hundred times, or despite the differences I got between each run of the library, the model was still very good at predicting toxic chemicals in particular. In terms of predicting interspecies variability, or intraspecies, I should say, that's left to be seen. I'm not sure. That would be something I think would be a really good idea to explore. As again, we're using an in vitro system, and it's going to be very limited in trying to account for that variability.
Yeah. A real quick question: are you planning to publish your results, specifically the experimental glue-gal data?
Yes, we absolutely are. I have a manuscript drafted already. We're just trying to, as I mentioned at the end there, trying to look at the versatility of the framework with a different chemical space. That's what we're currently working on. But we hope to release the experimental data for the entirety of the Pressbook library, and also the code around the active learning as well.
Okay, we have one more question if you're up for it.
Yeah.
We're getting a little bit over time, but the question is on, it's sort of following up on the cytotoxicity question. For the Tox-21 learning data, did you check the substance-specific cytotoxic burst or another cytotoxic assay battery to assure that the MMP hit calls in Tox-21 may not be attributable to false positive mitotox hit call by non-mitochondrial mechanisms?
So in that Tox-21 assay, so they ran mitochondrial membrane potential using mito-MPS dye, but also in the same wells, they then ran a cell titer glow. So the same assay I described using luminescence to measure cellular viability. So they had that internal control to determine whether it was cytotoxicity or mitochondrial toxicity. And where I gathered the data from was from PubChem. So this is published, that Tox-21 MMP data is published on PubChem, and PubChem assign activity scores based on this ratio between the MMP score and also the cytotoxicity score. So they only deem chemicals active that were positive in MMP and negative in the cytotoxicity.
Got it. Okay. Thank you very much. Thanks to both of our presenters, Tiago and Eliška, for giving us your work, sharing your work with us. Thanks to everyone tuning in for all of the questions and discussion. And we hope you have a good weekend. Take care, everyone. Thank you.